Welcome back to another explainer. So I want you to think for a second about the sheer massive volume of biological data driving modern science right now. I mean, every single day, laboratories worldwide are sequencing DNA, they're analyzing complex protein structures, mapping out these incredibly intricate metabolic pathways. It's literally an ocean of information. But, without a way to actually organize, search, and analyze all that information, it's just pure chaos. And that is exactly where biotechnology databases come into play. These are the highly organized digital repositories making modern biomedical science possible. They take millions and millions of disjointed data points and magically turn them into this incredible, searchable library of life. Okay, let's map out our route for today. First up, the biological data explosion. Second, organizing the bioinformatics library. Third, a tour of major databases. Fourth, real-world biological applications. And finally, number 5, challenges and future frontiers. So, kicking things off with Section 1, the biological data explosion. You've probably noticed that in recent years, high-throughput technologies and genomics have generated just an immense sea of biological data. I mean, this data has quickly become the absolute backbone of research across medicine, agriculture, microbiology, drug discovery, you name it. But to actually make sense of it all, we rely on a biotechnology database. Basically, this is a digital repository that stores biological information. We're talking sequences, structures, functional annotations, all lined up for research and analysis. It's the crucial tool that allows scientists to navigate the chaos, cut down on experimental redundancy, and share data globally in an instant. Moving right along to Section 2, organizing the bioinformatics library. Now, if we think of all this biological data as a massive library of life, you realize pretty quickly that you can't just dump millions of books in a giant pile on the floor and hope for the best, right? You absolutely need categories. So on one hand, we have primary databases. Think of these as the raw, unedited manuscripts of our library. This is the experimental data submitted directly by researchers, your pure DNA, protein, and nucleotide sequences. It's not heavily curated. But then, on the other hand, we have secondary databases. These are your polished, highly annotated encyclopedias. They contain analyzed, processed data that's been derived from those primary sources, which gives us things like structural classifications and really deep functional annotations. And actually, beyond just primary and secondary, we also have specialized databases. These act almost like dedicated reference sections in our library for highly specific research topics. So instead of housing broad, general sequence data, these focused digital repositories are entirely dedicated to a specific organism, maybe a specific disease, or a specific biological function. If you are researching, say, one highly specific genetic mutation, well, this is the exact room of the library you're going to go to. All right, let's get into Section 3, Tour of Major Databases. It is time to enter the VIP section of our biological library and check out the absolute heavy hitters. Let's just quickly walk through them. First up, we've got GenBank, which is maintained by the NCBI and handles our DNA and RNA sequences. Then you have UniProt. That's your go-to for highly curated, comprehensive protein information. Next is the PDB, or Protein Data Bank. This one gives us incredible 3D structures of proteins and nucleic acids, which, as you know, is absolutely vital for drug design. Then there's Ensemble, providing genome annotation and comparative genomics tools across tons of species. KEGG is super cool. It maps out complex biological pathways, including metabolic and disease pathways. And finally, PFAM, which classifies proteins into families and identifies conserved domains. Each one of these serves a fundamentally distinct and completely critical role in modern research. But hey, beyond those major players, researchers often need to rely on specialized databases for hyper-specific tasks. For instance, if a scientist is studying transcriptomics or gene expression, they're going to head straight to GEO or ArrayExpress. If they're researching genetic diseases, they'll lean on OMEM or DisGeNet. For drug discovery and pharmacology, that'll be DrugBank or CHEMBL. And for studying bacterial and viral genomes, they hit up the NCBI Microbial Genome Database or PATRIC. These highly targeted resources are what allow for those incredibly deep dives into specific subfields. Which brings us to Section 4, Real-World Biological Applications. So we spent a bit of time looking at the structural side of things, the how and the where. But what do we actually do with all this data? Like, why does categorizing protein families or RNA sequences actually matter to you and me? Let's pivot from the what to the why here, because these databases are quite literally the lifesavers of modern biology. Because we can read these biological books, we can apply that knowledge in some truly massive ways. In genomics, having this data enables rapid sequencing and comparative genomics. Over in drug discovery, it allows for virtual screening and target identification. I mean, we can literally look at drug-ligand interaction studies on a computer monitor before anyone ever sets foot in a lab. In agriculture, scientists are using this data for crop genome analysis to pinpoint disease-resistant genes. In medicine, it forms the foundation for cancer research and personalized medicine based on incredibly specific genetic disorder analysis. And in microbiology, it allows for rapid pathogen identification and tracking antibiotic resistance. The real-world impact here is just staggering. And finally, let's wrap up with Section 5, Challenges and Future Frontiers. Now, as incredible as this all sounds, managing global big data is definitely a double-edged sword. On the plus side, these databases provide easy access to massive datasets, saving just immense amounts of time and experimental cost. They support global collaboration and seriously enhance research accuracy. But, of course, we face real hurdles too. There's often a lot of data redundancy in some of these databases. Annotations can easily become outdated as new discoveries pop up. Data quality can vary wildly depending on the source. And actually integrating data across different databases, yeah, that's a major challenge that requires some seriously high-level computational skills. However, modern tech is actively upgrading our library as we speak. Just look at these recent advances. AI-based data annotation is swooping in to help researchers avoid drowning in information overload by automating the categorization process. Crowd storage and access mean a researcher sitting in Tokyo can instantly pull data that was sequenced just an hour ago in London. We're also seeing the massive rise of integrated multi-omics databases, real-time data updates, and wildly improved visualization tools. Essentially, technology is stepping up to solve the very limitations we just talked about. So if there's one key takeaway I want you to remember from this explainer, it is this. Biotechnology databases are structured repositories of biological information that support research in genomics, proteomics, drug discovery, and modern biomedical sciences. That quote really just perfectly cements the whole idea we've been exploring today. These repositories, they aren't just filing cabinets. They are the invisible, intelligent engines that are driving all modern scientific discovery. Which leads us to a really fascinating final thought. Future developments are pointing straight toward fully integrated global biological databases and super advanced multi-omics platforms. So I want to leave you with this. As AI fully integrates all of this global biological data, and as these real-time disease tracking systems become even more intelligent, how quickly will personalized genomic medicine become your everyday reality? The blueprint is already right there in the database. Thanks so much for joining me on this explainer. Keep asking questions, and I'll catch you next time.